What if aging didn't have to mean losing who we are? What if some minds could grow older without growing dim? Today, we explore that mystery through the work of neuroscientist Andrew Huberman, who helps us understand how certain brains resist time itself. In this talk, we uncover the science of superagers and the remarkable neural choices that keep memory, curiosity, and vitality alive. Aging is often assumed to come with inevitable memory loss, slower thinking, and cognitive decline, but the brains of superagers challenge this belief entirely. Superagers are older adults, typically over the age of 65 or 70, whose memory and cognitive abilities rival those of people decades younger. Rather than merely aging well, their brains seem to resist aging altogether, especially in areas responsible for memory, attention, and learning. From a neuroscience perspective, the most striking feature of superagers is the preservation of brain structure. Research using brain imaging shows that superagers retain greater thickness in the cerebral cortex, particularly in regions such as the prefrontal cortex and the anterior cingulate cortex. These areas are crucial for memory, focus, and decision-making. While most aging brains show gradual thinning in these regions, superagers maintain neural integrity, allowing them to recall information, recognize patterns, and stay mentally agile. Memory, especially episodic memory, the ability to remember events, experiences, and details from daily life, is where superagers truly stand out. In typical aging, the hippocampus, a brain structure essential for forming new memories, tends to shrink. However, studies reveal that superagers show little to no hippocampal shrinkage. This structural preservation translates into functional strength. They remember names, conversations, and experiences with surprising clarity. Andrew Huberman's work emphasizes that brain aging is not solely dictated by genetics. While genetics play a role, lifestyle and neural behavior significantly influence how the brain ages. Superagers often engage in consistent mental challenges, reading, learning new skills, problem solving, and staying curious. These activities stimulate neuroplasticity, the brain's ability to form and strengthen neural connections. When neural circuits are frequently activated, they are less likely to weaken over time. Another critical factor is attention. Superagers tend to exhibit strong attentional control, meaning they focus deeply and intentionally. Focused attention enhances memory, encoding the process by which experiences are stored in the brain. When attention is scattered or chronically distracted, memories form weakly. Superagers, whether consciously or unconsciously, optimize this process by being present and engaged, allowing memories to be stored more robustly. Emotional regulation also plays a key role. Chronic stress accelerates brain aging by increasing cortisol levels, which can damage neurons, particularly in the hippocampus. Superagers often demonstrate resilience to stress, either through social support, physical activity, or effective coping mechanisms. By keeping stress levels in check, they protect the very brain structures responsible for memory. Importantly, superagers are not defined by the absence of aging, but by resistance to cognitive decline. Their brains still age biologically, but at a slower rate and with greater efficiency. This reframes aging not as an unavoidable downhill process, but as a dynamic interaction between biology, behavior, and environment. Perhaps the most hopeful implication of superager research is that their traits are not exclusive or unreachable. While not everyone will become a superager, many of the neural habits that protect their memory, learning, movement, focus, emotional balance, and social engagement are accessible to most people. The science suggests that youthful memory is not just a gift of youth, but a skill that can be cultivated across a lifetime. In understanding superagers, we gain more than admiration. we gain a roadmap. Their brains remind us that aging does not have to mean losing our stories, our curiosity, or our sense of self. Instead, with the right neural choices, memory can remain a lifelong companion. Brain plasticity can persist across the lifespan for many years. Neuroscience promoted the idea that the brain becomes fixed after childhood, gradually losing its ability to change with age. Modern research, including insights shared by Andrew Huberman, has firmly overturned this belief. One of the most powerful discoveries in brain science is that neuroplasticity, the brain's ability to rewire, adapt, and form new connections, can persist throughout the entire human lifespan. This enduring plasticity is a key reason why some brains age more slowly than others. Neuroplasticity allows neurons to strengthen existing connections, weaken unused ones, and even form entirely new pathways. While it is true that plasticity is more pronounced in childhood, it never disappears. In adults and older individuals, plasticity becomes more selective, meaning the brain changes in response to meaningful, repeated, and focused experiences. Superagers benefit from this selective plasticity by continuously engaging their brains in ways that promote growth rather than decline. One crucial driver of lifelong plasticity is learning. When individuals challenge themselves to acquire new skills, such as learning a language, playing a musical instrument, or mastering unfamiliar technology, the brain is forced to adapt. These learning experiences activate circuits in the hippocampus and cortex, strengthening memory and attention networks. Over time, repeated activation builds cognitive resilience, allowing the brain to maintain sharpness even in later decades of life. Attention plays a central role in enabling plasticity. The brain does not change simply because an experience occurs. It changes when that experience is attended to. Focused attention releases neuromodulators such as acetylcholine and dopamine, which signal the brain that something important is happening. These chemicals act as gatekeepers for plasticity, allowing neural circuits to be reinforced. Superagers often exhibit strong attentional habits, which help their brains encode experiences more effectively and adapt more efficiently. Sleep is another essential factor in sustaining plasticity across the lifespan. During deep sleep, the brain consolidates learning by stabilizing new neural connections formed during waking hours. Poor sleep disrupts this process, weakening memory and slowing neural adaptation. Research consistently shows that older adults who maintain high-quality sleep preserve better cognitive function, reinforcing the idea that plasticity depends not only on learning, but also on recovery. Physical movement further enhances neuroplasticity. Exercise increases blood flow to the brain and stimulates the release of brain-derived neurotrophic factor BDNF, often described as fertilizer for neurons. BDNF supports the survival of existing neurons and encourages the growth of new synaptic connections. Regular physical activity, especially aerobic exercise, has been linked to improved memory and slower cognitive aging. Importantly, emotional states influence the brain's capacity to change. Chronic stress suppresses plasticity by elevating cortisol levels, which interfere with neural growth. In contrast, positive emotional engagement, curiosity, and motivation enhance learning and adaptation. Superagers often display a sense of purpose and engagement with life, creating a neurochemical environment that supports ongoing brain change. The persistence of plasticity reshapes how we think about aging. Cognitive decline is not simply a matter of time passing. It is strongly influenced by how the brain is used. When neural circuits are repeatedly engaged, challenged, and supported, they remain functional and adaptable. Plasticity becomes the brain's defense against aging. Ultimately, lifelong brain plasticity offers a hopeful message. Aging does not mean the end of learning, growth, or mental flexibility. Instead, the brain remains responsive to effort, attention, and care. By understanding and applying the principles of neuroplasticity, individuals can actively participate in shaping how their brains age, proving that change is not only possible, but powerful at any stage of life. Lifestyle strongly influences neural longevity. The way the brain ages is not determined by time alone. It is deeply shaped by daily lifestyle choices. Neuroscience research, including perspectives highlighted by Andrew Huberman, shows that habits related to movement, nutrition, sleep, and mental engagement can either accelerate neural decline or protect the brain for decades. Superagers provide compelling evidence that a brain can remain resilient and youthful when lifestyle consistently supports neural health. Physical activity is one of the most powerful lifestyle factors influencing brain longevity. Regular exercise improves blood flow, oxygen delivery, and nutrient supply to the brain. It also stimulates the release of brain-derived neurotrophic factor BDNF, which supports neuron survival and synaptic growth. Aerobic exercise, such as walking, swimming, or cycling, has been shown to preserve hippocampal volume, a critical region for memory. Superagers often maintain active routines, not necessarily intense workouts, but consistent movement integrated into daily life. Sleep quality is equally vital for neural longevity. During deep sleep, the brain clears metabolic waste products, including beta amyloid proteins associated with neurodegenerative diseases. Sleep also consolidates memories and stabilizes learning. Chronic sleep deprivation disrupts these processes, leading to impaired attention, memory loss, and faster cognitive aging. Superagers typically prioritize sleep, maintaining regular schedules that allow the brain sufficient time to repair and restore itself. Nutrition plays a foundational role in sustaining brain health. The brain requires a steady supply of energy, healthy fats, and micronutrients to function optimally. Diets rich in omega-3 fatty acids, antioxidants, and unprocessed foods support neural membranes and reduce inflammation. Excessive consumption of refined sugars and processed foods, on the other hand, contributes to insulin resistance and inflammation, which negatively impact cognitive function. While superagers may not follow rigid diets, their eating patterns often align with brain-supportive nutrition. Mental engagement is another crucial lifestyle element. Brains that are frequently challenged remain adaptable. Reading, problem-solving, learning new skills, and engaging in creative activities stimulate neural circuits and prevent cognitive stagnation. Unlike passive entertainment, active mental engagement forces the brain to work, strengthening memory and attention systems. Superagers tend to remain intellectually curious, treating learning as a lifelong process rather than something confined to youth. Social interaction significantly influences neural longevity as well. Meaningful relationships stimulate emotional and cognitive networks in the brain. Social engagement challenges memory, language, and empathy systems while also reducing stress. Loneliness and social isolation have been linked to increased risk of cognitive decline and dementia. Superagers often maintain strong social connections, whether through family, friendships, or community involvement, reinforcing both emotional well-being and brain health. Stress management ties all lifestyle factors together. Chronic stress elevates cortisol, which damages neurons and weakens memory-related brain regions. Effective stress regulation through mindfulness, physical activity, rest, or supportive relationships protects neural circuits. Superagers are not immune to stress, but they tend to recover more effectively, preventing prolonged exposure to harmful stress hormones. Importantly, lifestyle influences accumulate over time. Neural longevity is not the result of a single habit, but the compound effect of daily choices. Small, consistent behaviors shape the brain's structure and function across years and decades. This means it is never too early or too late to adopt brain supportive habits. The science of lifestyle and brain aging delivers an empowering message. While genetics set the foundation, lifestyle writes much of the story. By choosing movement, rest, nourishment, mental challenge, social connection, and stress balance, individuals can significantly influence how their brains age. Superagers remind us that a resilient brain is not accidental, it is cultivated, one habit at a time. Stress regulation protects cognitive function. Stress is an unavoidable part of life, but how the brain responds to stress plays a decisive role in how it ages. Neuroscience research, including insights emphasized by Andrew Huberman, shows that chronic, unmanaged stress is one of the most damaging forces to cognitive health. Superagers offer a powerful contrast. Their brains demonstrate an ability to regulate stress effectively, protecting memory, attention, and decision-making systems well into older age. At the center of the stress response is cortisol, a hormone released by the adrenal glands when the brain perceives a threat. In short bursts, cortisol is beneficial. It sharpens focus, mobilizes energy, and supports survival. However, when stress becomes chronic, cortisol remains elevated for long periods, leading to harmful effects on the brain. One of the regions most vulnerable to cortisol damage is the hippocampus, a structure essential for learning and memory. Prolonged exposure to high cortisol levels can shrink hippocampal volume, impairing memory formation and recall. Superagers tend to show lower baseline stress levels and faster recovery after stressful events. This does not mean they experience fewer challenges, but rather that their nervous systems return to balance more efficiently. This ability, known as stress resilience, reduces cumulative damage to neural circuits over time. Resilient stress responses protect not only memory centers but also the prefrontal cortex, which governs reasoning, impulse control, and emotional regulation. The autonomic nervous system plays a key role in stress regulation. It consists of the sympathetic branch, which activates the fight-or-flight response, and the parasympathetic branch, which promotes rest and recovery. Chronic stress keeps the brain locked in a sympathetic, dominant state. Superagers, however, often engage the parasympathetic system more effectively, allowing the brain and body to recover. Practices such as controlled breathing, physical movement, and mindfulness activate this restorative pathway. Sleep is closely intertwined with stress regulation. Poor sleep increases stress sensitivity and elevates cortisol levels, creating a harmful feedback loop. Conversely, quality sleep strengthens emotional regulation and reduces stress reactivity. Superagers typically maintain consistent sleep patterns, which helps stabilize stress hormones and supports cognitive performance. Social connection also acts as a powerful buffer against stress. Human brains are wired for connection, and meaningful relationships reduce perceived threat and emotional load. Positive social interactions stimulate oxytocin release, which counteracts cortisol and calms the nervous system. Superagers often maintain strong social bonds, giving them emotional outlets that reduce chronic stress and support brain health. Physical exercise serves as another effective stress regulation tool. While exercise temporarily increases stress hormones, it ultimately trains the brain to handle stress more efficiently. Regular movement improves mood, reduces anxiety, and enhances cognitive flexibility. Over time, physically active individuals show healthier stress responses and better preservation of cognitive function. Importantly, stress regulation directly impacts attention and decision-making. Under chronic stress, the brain shifts resources away from higher-order thinking towards survival mechanisms. This leads to reduced focus, impaired memory, and emotional reactivity. Superagers, by managing stress effectively, preserve the brain's capacity for thoughtful decision making, and sustained attention. The protective effect of stress regulation reframes cognitive aging. Memory loss and mental decline are not solely consequences of aging, but are strongly influenced by prolonged stress exposure. By learning to regulate stress, individuals can slow cognitive aging and protect essential brain structures. Ultimately, stress regulation is not about eliminating stress, but about mastering recovery. Superagers demonstrate that a calm, adaptable nervous system is a powerful shield against cognitive decline. Their brains remind us that protecting mental function begins with protecting the brain from the invisible wear and tear of chronic stress. Social connection strengthens aging brains. Human brains are not designed to function in isolation. From a neuroscience perspective, social connection is not a luxury but a biological necessity, especially as the brain ages. Research highlighted by Andrew Huberman and other neuroscientists shows that strong social bonds play a critical role in preserving cognitive function. Superagers consistently demonstrate rich social engagement, suggesting that meaningful relationships are a powerful protective factor against brain aging. Social interaction activates multiple brain regions simultaneously, including areas responsible for language, memory, emotional processing, and executive function. When people engage in conversation, they recall information, interpret facial expressions, regulate emotions, and respond thoughtfully, all complex cognitive tasks. This constant activation acts as a form of mental exercise, strengthening neural circuits and maintaining cognitive flexibility. Over time, socially engaged brains remain sharper because they are regularly challenged in dynamic, real-world ways. One of the most important ways social connection benefits the is is through stress reduction. Loneliness and social isolation are associated with elevated cortisol levels, which, when chronic, damage brain regions such as the hippocampus and prefrontal cortex. In contrast, supportive relationships reduce perceived stress and increase feelings of safety. Positive social interactions stimulate the release of oxytocin, a hormone that calms the nervous system and counteracts the harmful effects of cortisol. Superagers often maintain close relationships that help regulate stress and protect cognitive health. Social connection also influences brain aging through emotional regulation. Engaging with others provides opportunities to share experiences, process emotions, and gain perspective. This emotional exchange strengthens neural pathways involved in empathy and self-control. Older adults with strong social ties tend to exhibit greater emotional stability, which supports attention, memory, and decision making. Superagers frequently show high emotional intelligence, allowing them to navigate social situations with ease and resilience. Memory, in particular, benefits from social engagement. Conversations often involve recalling past events, names, and shared experiences. This repeated retrieval strengthens memory circuits and improves recall ability. Storytelling, reminiscing, and social learning all reinforce hippocampal function. Socially active older adults consistently outperform socially isolated peers on memory tests, reinforcing the link between connection and cognitive preservation. Social roles and a sense of belonging further contribute to neural longevity. When individuals feel needed, whether as friends, mentors, family members, or community participants, their brains remain engaged with purpose-driven tasks. Purpose activates motivation and reward circuits in the brain, enhancing dopamine signaling, which supports learning and plasticity. Superagers often remain involved in communities or family structures that give them a continued sense of relevance and meaning. Technology, mediated social interaction can also play a supportive role when in-person contact is limited. While not a full replacement for face-to-face connection, Regular communication through calls or video chats still engages social and cognitive networks. The key factor is consistency and emotional significance, not merely the mode of interaction. Importantly, social connection protects against cognitive decline at a population level. Studies consistently show that social isolation increases the risk of dementia and accelerated brain aging. Conversely, maintaining strong relationships correlates with slower cognitive decline and greater brain volume in aging populations. Superagers embody these findings, showing that social richness supports neural resilience. The strengthening effect of social connection reframes how we think about healthy aging. Protecting the brain is not only about individual habits, but also about relational health. Conversations, shared laughter, emotional support, and community engagement all act as neural nourishment. In the end, superagers teach us that the brain thrives on connection. Aging brains do not weaken simply because time passes. They weaken when deprived of meaningful interaction. By nurturing relationships and staying socially engaged, individuals can strengthen their brains and preserve cognitive vitality throughout life. The brain is built to learn, adapt, and evolve across the lifespan. Neuroscience research, including insights shared by Andrew Huberman, shows that consistent learning is one of the most effective ways to preserve neural circuits and slow cognitive aging. Superagers exemplify this principle by remaining mentally active and curious well into older adulthood, proving that learning is not confined to youth but is a lifelong process that sustains brain health. Learning activates widespread networks in the brain, particularly in the hippocampus and prefrontal cortex. These regions are responsible for memory formation, attention, planning, and decision-making. When individuals repeatedly engage in learning, these neural circuits are strengthened and maintained. In contrast, when the brain is understimulated, unused connections weaken over time, leading to cognitive decline. Superagers continually challenge their brains, keeping these circuits active and resilient. Novelty plays a crucial role in effective learning. Repeating familiar tasks does not engage the brain in the same way as encountering new information or skills. Learning something unfamiliar, such as a new language, musical instrument, or complex hobby, forces the brain to form new connections and adapt existing ones. This process enhances neuroplasticity and helps preserve cognitive flexibility. Superagers often seek novelty, approaching life with curiosity rather than routine. Focused attention is essential for learning to translate into lasting neural change. The brain does not strengthen circuits simply by exposure. It requires intentional engagement. When attention is directed toward a task, neuromodulators such as dopamine and acetylcholine are released, signaling the brain to reinforce relevant pathways. Superagers often demonstrate strong attentional control, allowing learning experiences to leave lasting imprints on the brain. Mistakes and effort are also vital components of learning. Neuroscience shows that the brain changes most when effort is high and errors occur. Struggling with new material activates error detection systems, prompting the brain to adapt and improve. Superagers tend to embrace challenge rather than avoid it, viewing difficulty as a natural part of growth. This mindset encourages continuous neural development. Learning is closely tied to memory preservation. Each new skill or concept requires encoding, storage, and retrieval, exercising memory systems regularly. Over time, this repeated use strengthens memory circuits and improves recall. Social learning, such as discussions or teaching others, further enhances this effect by requiring articulation and retrieval of knowledge. Superagers often remain engaged in conversations, mentoring, or intellectual exchanges that reinforce learning. The benefits of consistent learning extend beyond cognition into emotional and psychological health. Learning stimulates reward pathways in the brain, creating a sense of accomplishment and motivation. Dopamine release associated with learning enhances mood and reinforces engagement. This positive feedback loop encourages continued mental activity, which further protects neural circuits. Importantly, consistent learning supports cognitive reserve, the brain's ability to compensate for age-related changes or damage. A brain with strong, diverse neural networks can adapt more effectively when faced with challenges such as injury or disease. Superagers often exhibit high cognitive reserve, allowing them to maintain function even when some neural decline occurs. The preservation of neural circuits through learning reshapes the narrative of aging. Cognitive decline is not an unavoidable consequence of growing older, but a reflection of how the brain is used. When learning becomes a daily habit, neural circuits remain active, adaptable, and efficient. Ultimately, superagers demonstrate that learning is a form of brain maintenance. Just as muscles stay strong through regular use, neural circuits remain healthy through consistent challenge. By committing to lifelong learning, individuals can preserve mental sharpness, adaptability, and cognitive vitality, ensuring that the brain continues to grow even as time moves forward. As we reflect on the science of superagers, one truth becomes clear: the brain is not a passive victim of time, but an active participant in how we age. Through purposeful habits, learning, connection, movement, rest, and stress regulation, we shape our neural future every day. Andrew Huberman's insights remind us that longevity of the mind is not about avoiding age, but about engaging fully with life. If we nurture our brains with intention and curiosity, aging can become not a decline, but a continuation of clarity, meaning, and mental vitality.